Hello everyone, we are Team 8 and our project of the course of bacteriology is focused on the proposal of using Pseudomonas aeruginosa as a biocontrol agent in agriculture to help protect crops from harmful fungi.
Introduction to Pseudomonas aeruginosa. Pseudomonas aeruginosa is a gram-negative bacterium, which means it has a thin peptidoglycan layer surrounded by outer membrane, giving it a pink appearance under gram stain. It's also rod-shaped and motile, meaning it can move things to its flagella. This bacterium is commonly known as an opportunistic pathogen, especially dangerous for people with weakened immune systems. It's responsible for infection in the respiratory and urinary tracts and often causes nosocomial or hospital-acquired infections.
Pseudomonas virulence factors include lipopolysaccharide (LPS), part of its outer membrane that triggers inflammation and helps the bacteria resist antibiotics. Type 3 secretion system, a structure that injects toxin proteins into host cells to disrupt immune response. Type 4 pili and flagella, these structures allow the bacteria to adhere to surface and move, making it easier to infect tissues. And the one of our project is biocyanin. It has redox active properties that generate reactive oxygen species, or ROS, which can damage other cells.
The proposed mechanism of action is that by having a low molecular weight, biocyanin can easily diffuse through the cell membrane, and as a redox-active biomolecule, it can generate oxidative stress by increasing the intracellular levels of reactive oxygen species like superoxide and hydrogen peroxide, by disrupting cellular redox balance. This occurs through its interaction with intracellular pathways, specifically through the oxidation of glutathione to glutathione disulfide and the reduction of NADP plus to NADPH. The oxidative stress in bacterial, fungal, and mammalian cells causes several negative effects, including inhibition of essential enzymes, DNA damage, disruption of membrane potential, oxidative damage of the components involved in cell cycle and its regulation.
Previous solutions and their drawbacks. To solve this issue, chemical antifungals are commonly used. Unfortunately, these aren't sustainable solutions. They can harm beneficial soil organisms and pollute the environment. Fungi can also develop resistance to these chemicals, making them less effective over time. This leads us to explore safer, eco-friendly alternatives like biological control agents.
Our proposal is to use biocyanin as a biocontrol agent by introducing genetically modified Escherichia coli that produce biocyanin. We could target and inhibit pathogenic fungi directly in the soil. This would reduce the need for chemical pesticides and antifungals, helping to prevent resistance development and creating a more sustainable agricultural practice.
So, how would biocyanin become a biocontrol agent? In our plan, we'd modify an Escherichia coli to produce biocyanin by inserting key genes from Pseudomonas aeruginosa with a plasmid. The biocyanin producing Escherichia coli would then be applied to soil samples with pathogens. Once in the soil, the biocyanin would produce reactive oxygen species, causing oxidative stress in the fungal cells and inhibiting their growth. This would protect the crops without harming beneficial organisms.
The problem we are addressing is the widespread damage to crops from soil-borne fungi. These pathogenic fungi in agricultural soils can harm crops, leading to yield loss and economic impact. There are some statistical data that prove that it's a problem and its impact in the population. Agricultural fungal infections causes massive crop losses globally, affecting key crops like cereals, vegetables, and legumes. In countries like Mexico, the use of pesticides and antifungal chemicals is increasing to combat these pathogens. However, these chemicals can harm beneficial organisms and contribute to the development of resistance in fungi. In this graphic, we can see the frequency of the use of plaguicides reported in the period of 1980 and 2018. This information is according to the analysis of a study about the use of plaguicides in Mexico. For the methodology to the following our proposal, we will study soil and therefore crops like rice. The general materials and regions we need are genetically engineered Escherichia coli with a plasmid for pyrrhizamine synthesis. Soil samples inoculated with a mix of common soil-borne pathogens such as Fusarium oxyporum fungus and Rastoltonia solanacearum bacterium. Nutrient media for bacterial growth. Biosynonym detection kits, for example, for quantifying ROS levels in soil, and equipment for pH, temperature, and moisture control. We will use two groups of soil samples. The control group will contain soil with pathogens but no biocyanine, while the experimental group will contain both pathogens and biocyanamide producing E. coli. For the preparation, we need a culture that genetically modified E. coli to ensure biocyanine production. To express biocyanamine in E. coli, we have some key genes from the PHZ. There are PHZM and PHZS. The operon will be cloned and inserted into a plasmid vector. For that, we need to choose a plasmid vector that is compatible with E. coli and Pseudomonas, and that is a high spectrum expression vector. We can choose PBBR1MCS, that is a plasmid that replicates in different bacteria. Then we isolate the genes amplifying the PHZ operon genes from Pseudomonas aeruginosa using PCR. Then we insert the genes into the plasmid with restriction enzymes and DNA ligase to insert PHZ genes into the plasmid vector downstream of the promoter. Then we transfer E. coli with the constructed plasmid using heat shock or electroporation. After that, we place the transferred cells on antibiotic-containing media to select for E. coli cells that have taken up the plasmid. Then, we verify if it was a successful insertion of the genes by performing colony PCR or sequencing the plasmid from transferred colonies. After that, we culture E. coli and induce biocyanine production of monitoring biocyanamide producing using colorimetric assays of spectrophotometry.
We expect that our approach will decrease pathogen levels in the soil and improve crop health without the negative effects of chemical antifungals. Our methods would offer a sustainable, eco-friendly alternative to synthetic chemicals, ultimately leading to healthier crops and reduced environmental pollution.
Application and monitoring. Applying the biocontrol agent to the soil will involve infusing soil granules with concentrated pathogen and bacterial suspensions, ensuring even distribution. We'll also monitor ROS levels to confirm the activity of biocyanine. Our main goal is to observe reduced pathogen density and better crop growth in the experimental group.
We've supported our project with papers and books on Pseudomonas aeruginosa, agricultural impacts of soil pathogens, and potential of microbial bioformulations in sustainable agriculture. Thank you for your attention.